This is the Biology Paper 1 Foundation paper, right, 2023. Right, question number 1. So what I'm going to do is I'm going to go through the answers and a bit of theory that then goes with it. The human body is made up of different types of cell. Draw one line from each type of cell to the organ system where that cell is found. Okay, so a brain cell. Okay, so what you've got to do on this one is you've got to get a ruler out and you've got to do it. Don't do funny little squiggly lines. Now I haven't really got a ruler. I'll try and do it straight. Nervous system. A red blood cell, so that's not reproductive. It's not respiratory because that's breathing. That is then the circulatory system. And the third one, sperm cell, that's not respiratory because that's breathing. So that is then reproductive system. Explain one way a sperm is adapted to its function. Okay, look at the answers here. Explain. So what you've got to do is you've got to give sort of like a reason, a describe, and then explain how it actually does it. So it has a tail. Okay, so that is then my kind of first mark, which is tells me I describe it's got a tail, so it can swim. So what I've got then is so is like a key word that comes into it. Sometimes it's because so works very well. Now it only says one example, okay, but what I could do then is I could say it has many mitochondria. So it can release more energy from respiration. Now, one other little thing that may well help here a little bit is many mitochondria. It would be good to write in there sperms have. Sperms have. Okay, so what you're doing then is you're making it as obvious as possible what the answer to the question may well be. Now, there are other answers. So, for example, a sperm is streamlined so it can swim faster. Okay. Figure 1 shows a cell. Oxygen molecules are shown inside and out of the cell. So those little dots there, right, are the oxygen. Now I can see immediately that what I've got there is I've got more outside than I've got inside. Give one way in which you can tell that figure 1 is not a plant cell. Okay, so not a plant cell. So if I think there's 5 things that make up an animal cell, and there's 3 that make up or extra things that are in a plant cell. So there's 5 in an animal, 3 in a plant cell that are different. Okay, give one way you can tell it's not a plant cell. Okay, so I could say it does not have a vacuole. You could put permanent vacuole. It does not have a cell wall. Okay, and it does not have chloroplasts. Okay, so any of those 3 would then work, but just make sure you're writing it as clear as you possibly can when you're doing it. Question 1.4. Which part of a cell controls movement of substances in and out of a cell? Okay, so there's my cell, there's my nucleus. Right now, the nucleus, it contains the DNA, so it's definitely not that job. Cytoplasm, which is that one there, right, is then where all the chemical reactions happen. So tick one box, cell membrane. 1.5. What is the name of the process that moves oxygen molecules into the cell? Okay, process, that's telling me an answer. Oxygen molecules is telling me then it is a different answer. Okay, well it's kind of on the same sort of pattern. So the processes are active transport, diffusion and osmosis. So what I've got to do then is I've got to think about which one is it likely to be. Okay, it's not osmosis because what it's doing is it's nothing to do with water. Is it active transport or diffusion? So I've got to remember, right, so active transport is then low to high, okay, and this isn't. It is high to low. So the answer to that one is diffusion, okay, and then the reason for it is that the concentration of oxygen is higher outside of the cell. Okay, And we know diffusion is the movement of particles from a high concentration to a low concentration. So therefore it is definitely diffusion. Okay, name 2 substances that move into most cells in the body from blood. Do not refer to oxygen, right? From the blood into the body, right? So what does a cell need? It needs glucose. Okay, so that is going from the blood into the body. It needs water. It needs amino acids. I shouldn't write it small really. It needs fatty acids. It needs glycerol. Okay, it needs minerals. Okay, now there's one little question on there, one little part. Name 2 substances that move into most cells. Okay, so if you think about it, I'm going to do this in red. You could have written CO2, right? But CO2 moves out because that is a byproduct, okay, of the respiration process. Ooh, that's a bit small. All right. Enzymes break down food in the digestive system. Figure 2 shows the human digestive system. The enzyme amylase digests starch. Which part of the digestive system produces amylase? Right. So what I've got there is I've got A is the liver, B is the stomach, and C is the small intestine. Now I know liver's job is to produce bile, so it's definitely not A. I know the stomach's job is mainly to sterilize food, and it also has pepsin in it, which is protein, so it's not B. So the answer is C. Now don't do your big crosses like that on your exam, okay? I'm just doing that as an example, right, of kind of the train of thought. Okay. What molecules are produced when starch is digested? Okay, so starch is a carbohydrate. So therefore, what it is, it's a big long chain of something. Amino acids is a big long chain, which makes up proteins, so it's definitely not that. Fatty acids, right, that's the breakdown of fats. Got fatty acids and glycerol, so it's not that. So what's produced? Sugars. Okay, in particular, if that breaks broken down there, it could be glucose. Where is digested food absorbed into the blood? The liver's job is pretty much in the digestive system to produce bile. The pancreas produces enzymes. The small intestine's job is to absorb food into the blood. A student investigated the effect of pH on the digestion of starch. pH. Starch and amylase. Okay, so amylase is an enzyme. And I know it's an enzyme because it ends in ASE. This is the method used. Put 1 drop of iodine solution into each well on a spotting tile. So let's draw a spotting tile. There's a spotting tile. Well, there's the little dimples. Right, very, very badly drawn. So what I do is I'm going to put iodine. So iodine is kind of like an orangey brown colour, so I've almost got the right colour. Prepare my amylase solution at pH 5. Okay, so there's a boiling tube, and what I've got in there is I've got my amylase, and it's at pH 5. Mix the amylase solution with starch solution in a test tube. Mix. Okay, so I've got starch solution and I mix it. Every 30 seconds, so what I've done is I start my stop clock. Every 30 seconds, remove a drop of the amylase. So then what I've got to do is I've got to move some of this. Okay, remove a drop of the amylase starch mixture, add into each drop to the iodine solution in a different well. So I'll take a little drop of that and I put it in there every 30 seconds. And obviously you've got to remember that you did it with 0 seconds as well. Record the colour of the iodine after the amylase starch mixture has been added. Repeat steps 2 to 5 for amylase solutions at different pHs. What was the independent variable? Don't mess in class. So dependent measure, independent, what am I changing? So I'm changing the pH. Okay, that's what I'm doing, right, in between each one. I start with pH 5, and then what I then do is I then work my way along it. Figure 3 shows the results on the spotting tile. Fabulous, it's a bit different to the spotting tiles that I've seen. What colours do the symbols in the key represent? Choose answers from the box. So black represents black. Okay, so that is then when the iodine reacts with the starch and it actually goes black. Right, sometimes I say blue-black, I'm not quite sure what it is, it looks black to me. White is then when there is no reaction. So therefore, if there is no reaction, it is orange, which is kind of the original colour of the iodine itself. Okay, so if I could do some colouring in, orange, fabulous, orange, orange. So that is then where, oops, missed, that is then where the starch has already been digested. Okay, look at the results for pH in figure 3. Okay, pH 6. Okay, so there's pH 6. Okay, so I can look all the way along and I can still see that there is starch present there. So therefore, how many minutes did it take for the starch to be digested? It will be, let's do an orange there, it will be 3 minutes. What was the optimum pH for amylase to work? So that basically means optimum is the quickest. So the quickest is then there, pH 7. Plants need water for photosynthesis. Okay, we know the photosynthesis equation. Where do plants obtain water for photosynthesis from? Okay, now this actually caught me out, this one. All right, and what I did is I wrote down roots. Okay, but that isn't where they get the water from, right? They don't get the water from the roots. What they do is they get it from the soil. Okay, and that actually caught me out when I did it. Plants lose water from their leaves through small pores called stomata. What is the evaporation of water from leaves called? Okay, evaporation of water from the leaves, active transport, no. Respiration happens in cells, transpiration. Transpiration is then where water, I guess it's like the plant version of sweating. A student investigated the mass of water lost from a plant. Figure 4 shows the apparatus. Okay, there's a plant, okay, plastic bag around it, and there's the mass balance, and it says 510.7 grams. This is the method used. Seal a plastic bag around a pot of a potted plant. Place the plant on a balance in a room at 20 degrees C. Record the mass every 5 hours. Calculate the total mass of water lost from the plant after each hour. Okay, so they're the results. Okay, so at time 0, 510.7, mass lost is there. 5.3 is then calculate mass X in the table. So calculate mass X. So if I've got mass X, okay, which is that one there, okay, so if I start at 510 and I've lost 9, then what my sum will be, it will be 510.7, take away 9 equals 501.7 grams. And make sure you write it in your little line here, 501.7 grams. What was a graph? Cool. Right, total mass of water lost in grams against time in hours. Very interesting. Okay, so that means as the time increases, the total mass of water lost in grams increases, and it's a lovely straight line as well with no little anomalous results, which makes it interesting. Okay, what's over my face there? That's not good. What was the rate of water loss from the plant? Okay, so we're looking at per hour. Okay, so if I go time in hours, and I go from 1 hour using my lovely straight ruler that I don't have. Okay, oh, look how wonky that is. Okay, over to there, and then so that's my time in hours, and then I go across to the mass of water lost. So how much do I lose per hour? 1.8 grams per hour. Okay. 3.5. Investigation was repeated at a lower temperature. Draw one line on figure 5 to show how the results would be different at a lower temperature. Okay, so logically, right, what we've got then is we've got lower temperature, therefore there's going to be less water lost. So total mass of water lost in grams is then going to be less. So what I'm going to do is I'm going to get my lovely straight ruler out that I had before, okay, and I'm going to start at the same point because that's the time when it starts, and then I'm going to draw a line lower, and I'm going to stop it there, okay, and that is a lovely straight line done with a ruler, okay. That then answers that question. 3.6. Suggest one change to the investigation would increase the rate of water loss. Do not refer to temperature. So what you could do is you could talk about air movement. Okay, you could talk about light intensity. You could talk about humidity. But you know what? I've just answered that question there and I've got no marks for it, because I've got to talk about how I would increase the rate of water loss. If I just put down air movement, it's right, but that's not telling me how I would increase the rate of water loss. So for air movement, I need to increase air movement. So that's windier. If it's windier, more water gets taken away. Increase light intensity. So if there's more light, it's going to lose more water. Decrease humidity, because the more humidity, the more moisture in the air already, the lower the amount of water lost through transpiration. Okay, so make sure, even though it's only one marker, right, make sure what you're doing is you're telling it as exactly as you possibly can. Ooh, it's the heart. I like the heart. The vena cava carries blood into the heart from the body, right? So let me think about it. Right atrium, right ventricle. Okay, so I think that there is the vena cava, because what it's doing is bringing blood into the right atrium from the body. Then what it does is the blood then comes in through the right atrium, through there, and then it goes up the middle. So that must be the pulmonary artery going out at B. Then what it does, it goes to the lungs, and then what it's got to do is then got to come back into the left atrium. So there, so it's going to go from there, it's going to go around to the lungs and back to there. So that is then the pulmonary vein. Then what happens is it goes through the left atrium, through the left ventricle, up there, and I think C is then the aorta. And then what it does, it goes around the body, and eventually what happens, it comes back through the vena cava. Right, which blood vessel in figure 6 is the vena cava? Vena cava. Okay, so that is A. Okay, I've just kind of worked it all out. Okay, which chamber of the heart pumps blood to the body? So which chamber? So if you think about the diagram we just had, okay, so that'll be, it's on the right hand side, so therefore it is the left ventricle. What is the name of the blood vessel that carries blood to the heart muscle? So we're not talking about anything else, we're just talking about to the heart. So it's not the aorta, because that's all around the body. It's not pulmonary artery, because that's the lungs. Coronary means heart. The heart and some blood vessels contain valves, correct. Which type of blood vessel has valves? It's not artery, because that's under high pressure. Capillaries are way too small to have valves, so it's veins. What is the function of a valve? Okay, so what you have is, so that's, let's say that's your leg, that's a vein in your leg, right? And what happens is you have to look at little pocket valves. So when the blood, let's do it in red, that makes sense. So when the blood goes up, what happens is it doesn't have quite enough energy and force to get back. So what it does, it kind of drops down, and the little valves flatten out to stop the blood then going back down. So the function of a valve is to prevent backflow of blood. Figure 7 shows a capillary. Explain one way the capillary is adapted for its function. Explain. So that means we're going to be doing a because or a so. Okay. Walls are one cell thick. So, let's circle that in purple. So, there is a short diffusion distance. Okay, that's good. Okay, maybe another one. Let's go for capillaries have a large surface area. So there is a good exchange of substances. So in an explain question. 4.7. Okay, so table 2 shows information about the blood of 4 people. W, X, Y and Z. Red blood cells, white blood cells, platelets. Okay, so red blood cells, don't want yellow anymore. Red blood cells is carrying oxygen. White blood cells is infection. Platelets is clotting of blood. Person W has 5 million red blood cells in 1 millimetre cubed of blood. Now, that sounds like a lot. What is 5 million in standard form? Oh my God, 5 million, is that the right number of zeros? 3, 6 in standard form. Right, so what I'm going to do is I'm going to start here. There's my decimal point, and we're going to go 1, 2, 3, 4, 5, 6. So it's 5 times 10 to the 6. Tick. Okay, so draw one line from each description to the person in table 2 it describes. There's table 2 again. The person most likely to have an infection. That means they need lots of white blood cells. It's got to be person W. Straight line, use a ruler. Person whose blood will not clot properly. So not clot, that's platelets. So they haven't got very many platelets. That's 20,000 is X. 4.9. Explain, the greater the height above sea level, the less oxygen there is in the air. Okay? People who live high above sea level have more red blood cells than people who live at sea level. Okay? Some athletes train in mountains high above sea level. Okay, again, that makes sense, because then they'll have more red blood cells to carry more oxygen. Explain why having more red blood cells will improve an athlete's performance. So, firstly, more oxygen can be transported by or in red blood cells. Okay, so the whole point in it is explain why having more red cells will improve the performance. All right, so I've talked now about having more red blood cells is good. 2, because oxygen is needed for aerobic respiration. Okay, so I've talked about we need oxygen. Why do we need the oxygen? Is for aerobic respiration. And then number 3, so now I've got to answer the question here. It's about athletes' performance. So more energy can be released. Oh, breathing system, lungs. Okay, figure 8 shows the human breathing system, A, B, C, D. Okay, so A looks to me like the trachea, B looks like the bronchus, C looks like the bronchioles, and D looks like the alveoli. Name parts A and B. Okay, so A is the trachea, B is the bronchus. Where does gas exchange happen in the breathing system? Okay, so that is then D, which is then the alveoli. Should have had a picture up there really. Okay, we'll just scan back if you need to. 5.3. Give 2 ways the lungs are adapted for efficient gas exchange. Okay, 2 ways. Large surface area. Okay, so what I've done is I've said it's got a large surface area. I haven't just talked about surface area. They've got a good blood supply. Okay, so it's got a good blood supply. I don't just talk about blood supply. And there is a third one, which is the walls are 1 cell thick. Okay, now what it's actually doing that is just talking about 2 ways. It's just, it's not talking about an explanation. So you don't need to say large surface area, so, or good blood supply, so, right, there's a diffusion gradient, right, or a concentration gradient rather, or 1 cell thick, so there's a short diffusion pathway. You don't need to do that. Table 3 shows the percentage of gas in air breathed into the lungs and air breathed out of the lungs. So in, out. 5.4, okay, explain the differences in the air breathed into the lungs and the air breathed out of the lungs. Explain. So what I'm going to go for, I'm going to go for describe and then a because or a so. So here we've got less oxygen in exhaled than inhaled because it's used for respiration, because it is used for, right over here, respiration. Okay, got him right. Okay, also there is more CO2 in exhaled. Okay, so what I'm doing is I'm making sure I tell you whether it's exhaled or inhaled, right? Because CO2 is a product of respiration. Okay, so what I'm doing here is, if you look at it now, right, let's get yellow, because. Okay, so think about it. An explain question is because or a so. Let's do red. There is no difference between nitrogen inhaled and exhaled as not needed for respiration. Okay, so what I've done there, as needed, I could have again because it's not needed for respiration. 5.5. The percentage given in each column of table 3 do not add up to 100%. Right? Okay, so they don't add up to 100% because there are other gases not named. So for example, argon isn't on there. And also it could be because the numbers are rounded. Okay, so it's not 21.36, right? It's rounded. Okay, so question number 1 is, communicable and non-communicable diseases are major causes of ill health. Which disease is a non-communicable disease? So non-communicable, you cannot give it to somebody else. AIDS is a virus, yes you can. Gonorrhea you can, malaria you can, cancer you cannot pass on to anybody else. Okay, and again, make sure you only do one tick, and if you're unsure, just tick it anyway or have a random guess. Obesity is a factor or risk factor for many non-communicable diseases. Give one non-communicable disease that obesity is a risk factor for. Okay, so we're talking about somebody who is larger than they will be expected to be. Do not refer to the diseases given in 0.11. Okay, so that's cancer. So you can't talk about cancer. Right, now there's a number of these different ones that you could have. You could have coronary heart disease. Okay, that is then due to obesity. It's due to large levels of fat in the blood and in arteries. You've also got heart disease, okay, which is kind of another option, very similar to the previous one. You've got diabetes. You've got high blood pressure. Okay, and then you've even got something like depression. Okay, so they are all non-communicable, right, that are often due to being overweight. Question 1.3. National policies, right, so we're talking about policies, are used to help people who are obese to lose weight. Okay, so it's like a government thing that's been introduced to help people actually lose a bit of weight. One national policy is reduce the amount of sugar added to food and drinks. Suggest one other national policy. So they're not after a theory, what they're after is they're after a policy that could be put into place to help people lose weight. So you could have tax on sugar, okay, sugar tax, or a fat tax, okay, where foods that then have got more sugar or more fat in right, then what you can do, the government could add a tax to it. There could be a policy on education. You could have a policy where you are restricting advertising, okay, on foods that have got large amounts of sugar and fat in them. One that's quite a good idea as well, you could have calorie information in restaurants. Okay, so you know you go to McDonald's and you see on the back of the pack where it says there's this amount of calories in this, there's this amount of these, right? And generally in restaurants you don't do it. But what you need to do is you need to make sure you are talking about policy that the government could come in and they could then say this is a national policy that is then going to reduce the weight of people that are already obese. Question number 1.4. So what we've got here, BMI, body mass index, is a measure of obesity. BMI is a calculated using the equation there, right? So we've given the equation, which is fantastic. Then what we're given a table that tells us the category. Okay, so a person is 1.64 meters tall and has a mass of 69 kilograms. Okay, so I'm now going to write down the formula. BMI equals body mass in kilograms. So I'm going to look for my mass in kilograms, which is 69. You just got to do your calculations every single time. Divided by your height in meters, 1.64 squared. Okay, don't forget the little squared that's up there, because all of a sudden that would just be then throwing marks away. Then what I would do is I'd do equals, and I'd do my sum, and I'd go 69 divided by 1.64 squared, so you do your squared first, 2.6896. Now the reason why I do that is I do it as an extra step, is because you could quite easily just put into your calculator 169 divided by 1.64 squared, and the sum could work out wrong based on your calculator. That then says the answer is 25.6. Okay, so what you've got there is you've got a mark for your calculation, you've got a mark for your answer, and then the bit that people forget about is the person's BMI category, right? And often what people will do is they'll write in the number there. Right, it's not asking for that, it's asking for the category. Okay, so the category, you go over to this table on the left hand side here, you look at 25.6, which is in that one there, and so what you'll do here is you will write in overweight, okay, because that is then the category. Question number 1.5. Okay, so scientists investigated the effect of smoking and of BMI on the birth mass of babies. Women's BMI categories were determined before the woman became pregnant. Okay, so they're looking at women's BMI, pregnant women, and it is before women became pregnant. Okay, so 1.5 suggests why BMI categories were determined before the woman became pregnant. All right, so women gain weight during pregnancy. Okay, so you just got to think about it. You've got to think about a typical woman and their BMI. Right, their BMI might be completely and utterly normal, but when a woman is pregnant, what they will naturally do is they'll naturally put on a little bit of weight. Okay, and also suggests by BMI were determined before the woman became pregnant, and also BMI done before pregnant because the baby adds weight. Okay, so obviously if a woman's pregnant, all right, then the baby's going to grow inside the woman, and the woman's going to naturally put on weight as well. Okay, 1.6, right, let's have a look at our graph first. Figure 1 shows the results. Mean birth mass of baby in grams. Okay, so we've got to notice that that's in grams and it's the mean birth mass. BMI category of women before pregnancy. Right, so now what we're doing is we're looking at smokers and non-smokers. Right, so what we can do is we can see patterns. Okay, so immediately, right, I can see that all the white boxes are bigger okay, than the smokers' boxes, which means the mean birth mass of non-smokers is bigger than smokers. Okay, so give two conclusions, right? And it's numbered there, 1 and 2. So obviously what I can do then is I can then work that out. Two conclusions, right? So what I could do here is I'm not going to be able to write this very well. Smokers' babies have lower birth mass. Okay. Now what you could do there is you could have non-smokers' babies have a higher birth mass. Okay, you can flip it round either way. Okay, then what you could also do is you could also have that obese women have heavier babies, and you can also have underweight women, I can't write it all, underweight women have lighter babies. Okay, so you can see that because you've got an obese woman who's a non-smoker has a heavier baby than an underweight woman who is also a non-smoker. So there's a good comparison. We can also talk about smoking. Smoking had least effect on overweight. Oops, can't wait. Overweight people, and then you can do like converse, which is then smoking had more effect on underweight. Okay, now obviously what it's doing there is asking for 2. It's only asking for 2, right? But what I've done is I've given about 4 different possible examples just by looking at that individual graph there. Question number 1.7. Measles is a communicable disease, which means you can give it to somebody else. And it's also a virus. Okay? Describe how the measles virus is transferred from person to person. Okay, so it's a describe, so all we're doing is we're telling facts, but I do notice here it's worth 2 marks. All right, so just look at the question. Describe how measles virus is transferred from person to person. So if I've got measles, how do I get it to somebody else? So the first stage is I could cough, I could sneeze, and that could then give out droplets that have then got the virus in it. But if you think about it now, all I've done is I've then talked about how I actually get it out of me. I haven't talked about how I actually get it into the other person. Then the second one is the droplets are inhaled by someone else. Okay, so if you look at the question then, right, how is it transferred from person to person? Okay, it's not how I give it out, or it's how the other person gets it in, it's how it is actually transferred. Okay, 1.8. Athlete's foot. Athlete's foot is a communicable disease. Okay, again, it's a fungus. The athlete's foot fungus infects the skin on the feet. 1.8. Scientists estimate that 17%, that's obviously important, of the UK population have athlete's foot. It's quite scary really that 17% have got it all the time. The estimated UK population is that many, 67 million, etc., etc. Calculate how many people are estimated to have athlete's foot. Right, so what they're doing here, calculate. I'm going to be doing a sum. I'm going to do my working out here, and then what I'm going to do is I'm going to put an answer here. How many people are estimated to have athlete's foot? Now, there's a number of ways you can do a percentage. The way I like doing it is 17 over 100 multiplied by 679,61900. Or you could do 0.17 multiplied by 679,61900, whatever it ends up being. That equals 11,553,523. Okay, lots of numbers. Make sure, just like I was doing then, make sure you're checking your correct numbers. Estimate number of people with athlete's foot. You might leave your answer there, but don't. what you've got to do is you've got to make sure you write it on this line. There's no point in risking it even if you've got the marked answer up there. 11, 5, 5, 3, 5, 2, 3 is your answer. Now in the actual scheme of mark scheme, it actually says you can have the end of it as 500. Why would you do that? Right, you just write down exactly as the actual answer is. So just keep it exactly there. 1.9. Athlete's foot fungus grows in moist conditions. Lovely. Suggest one way a person could reduce their chance of catching athlete's foot. Okay, so think about where you're going to catch it. Right, you're going to probably catch it somewhere like a swimming pool. All right, so what you're going to do is you need to dry your feet properly. Don't share towels or socks. Not quite sure why you would share socks, right? But if you've got somebody in your family, right, they need to be checking to see if they've got athlete's foot or not. And if they have got athlete's foot, right, they need treatment for it. And you also don't share towels and socks. Okay, and what those people that have got it, what they can do is they can use fungicide. Okay, it's just a powder or a cream. Okay, and also if you've got it, or if you suspect you might have it, or somebody might have it, or just for purely prevention, right, just in case, right, don't walk around in bare feet. Okay, and obviously in your own home, that's fine, unless you know somebody's got it. So what we're thinking about is thinking about public places and contaminated places. Right, question number 2, right, 6-mark question, all right, and it looks to me like it's a little bit of a practical kind of thing. All right, so what I've got here is I've got a microscope view, right, of some cells. I've got a microscope, I've got a ruler, and I've got, oh, that's key, got a prepared slide of onion cells. So it's not telling me in this, then I haven't even read the question yet, it's not telling me in this how to actually prepare the slide. It's going to be asking me something to do with the slide that's already ready. Describe how the student could use the apparatus, right, we're talking about the apparatus, 1, 2, 3, excuse me, to estimate the mean length of an onion cells on the slide. Okay, so we've got to use the practical stuff here to calculate or estimate the mean length of onion cells on the slide, right? So I've got my 3 pieces of equipment, right? So what I'm going to do is I'm going to just do bullet points. First thing I'm going to do is I'm going to put my ruler on the stage of the microscope. Okay, so I'm using the ruler and I'm putting it on the stage of the microscope. Bullet point number 2, let's change colour. Okay, focus on the ruler. Focus on the ruler, and what you can actually do underneath there is you can see kind of the little dark, the black lines on it. Once you've focused on the ruler, you can then work out the diameter of the field of view, and that then needs to be in millimeters. Okay, I'll just do it in black. So what you've now got is you've now got the distance from there to there, because you've done it on your ruler, and you then know the distance from there to there in millimeters, which is great. Now focus on the cell, right? So what you do now is you focus on the cell. Okay, so you're not worried about the ruler anymore. All you want to do is focus on the cell because you now know the distance across. Use the same magnification. And then count the cells across. Okay, so it looks like it's 1, 2, 3. So if I've just got red here, I've got 1, 2, 3 cells across. Okay, that kind of works. It's right in the middle there. Then calculate a mean. All right, so what you'll do is you do the distance in millimeters divided by 3, which then will give you the mean length of onion cells on the slide. All right, but what you've got to do is you've got to think about the exact process. And because it's talking about apparatus, make sure you talk about all the different pieces of apparatus.